and welcome everyone to the second webinar out of the webinar series hosted by Advaita BioInformatics. Today's presenter is Andrew Olson, who is the Vice President for Business Development here at Advaita. Andrew will be walking you through uploading data into iPathwayGuide and the various data formats that you can use to upload any kind of data that you have into the application. After the presentation, we will answer any questions that you may have. Please use the chat box of questions to type in any questions throughout the presentation. So with no further ado, here is Andrew.
Thank you, Sidra. Hi, everybody. Thank you for attending today's webinar. And we'll be talking about the different ways that you can get your data into iPathwayGuide. So the agenda, just very briefly, will be just giving a general overview and talk about the different input data types, how to actually get that data into iPathwayGuide, and then we'll talk a little bit about single versus multi-contrast data sets. We'll even go into how you would upload and analyze AFI cell files, and then we'll just very briefly touch on how to construct a meta-analysis. We've got an upcoming webinar that'll go more in depth on that part of it, and then of course the Q&A session. So I'd like to always talk about some quotes that we get. So here's one that we recently received. A highly renowned collaborator proclaimed I had given him the bioinformatics analysis he had been hoping to get for 20 years or so. My aim is to provide a set of testable hypotheses to clients, not simply a list of genes. Your software fits that bill extremely well, in my opinion. This comes from Dr. Ben Harrison at the University of Louisville, from the bioinformatics core there. And it just is an affirmation of the high quality analysis that we've here at Advaita strive to provide all of our customers. So let's talk about what we're talking, or put this into context for today. Here's a typical bioinformatics pipeline where you have your instrumentation that produces some type of either transcript or proteomics data, and then they go through some type of alignment and annotation, and then you have your phenotype comparison where you're looking at your, say, your control versus your comparison, and you get your differential expression. Today, what we're talking about is how you take that data and update it and upload it into iPathwayGuide and perform a variety of analyses, not just pathway analyses. The one exception to that we do cover is in Affymetric cell files, we do the normalization and calculate the differential expression for your two phenotype comparisons. And I'll show a little bit more about that later on in the presentation. The general rules that you need to be aware of when working with iPathwayGuide is that we need three elements of, or three types of information. We need the gene symbol, the fold change for that for that symbol, and then the associated p-value. And we recommend using the adjusted p-values. The other element is that you should upload your entire list of measured genes or proteins, and that's so that we can assess the appropriate background and not just assume that it's whole genome or some subset. And before I forget, these slides are available to you, so you're welcome to download these. This is a nice chart of the different data types that we support here, and of course we have the AFI cell files at the top, and then the typical workflows for RNA-seq are CuffDiff and DE-seq. We will be adding EdgeR in our next release. We also support Lima if you're used to working with Bioconductor and the R package there. But this is also the workflow that's supported out of NCBI's GEO and their GEO to R tool, which will provide you with differential expression from within GEO. We support Jump Genomics, NSolver from NanoString Technologies. This is gaining a lot of popularity for targeted panels. And then on the proteomics side, we support SciEx SWATH data. It's actually SWATH 2.0 for their data independent acquisition data. And then, of course, if you have any kind of like Excel file or something like that where you have tab delimited with those three elements, the gene symbol, fold change, and then the p-value, we can support that. I have asterisked some of these formats with an asterisk, and that's to identify that those types of data can support multiple contrasts, and I'll explain what that means here in just a couple slides. So uploading data in iPathwayGuide is really simple and easy. Basically, once you've submitted your data, the only additional information that you need is to provide a title and a description, and then you have the option of updating your contrast labels and then also adjusting the thresholds. So let me show you very quickly how you do that. So from the iPathwayGuide dashboard, you would simply select Analyze New Experiment. You choose your organism, the file type. Here I think I'm going to use a Geo to R file. And then you choose your file. So in this case here, I'll select that appropriate file. And then you simply click Upload. And it depends on how fast your internet connection, but it usually just takes a few seconds to upload the data, and then we'll be able to see what's available to us. So as I stall here while it uploads, there we go. So we can provide it with a brief title and a description. And by default, you have condition versus control, but maybe I wanted to say treated and untreated. And then I can adjust my thresholds here as needed. The thing to read here is that we've identified 20,000 genes, more or less, and then the default thresholds, we use a 0.6 absolute fold change threshold, and then for the p-value, we have 0.05. And as a result of those thresholds, we have 2,129 DE genes. Now we can adjust those thresholds as needed, but let's just take a quick look at what the volcano plot looks like. And maybe we wanted to increase our fold change to maybe say 2.0. Just make sure you hit the view and apply to get that updated fold change threshold, and you can see that we've reduced the number of DE genes significantly here. We're now down to about 111. And I'll talk about what is the ideal thresholds to use in just a second. Once you're satisfied though, just make sure that you don't have any red boxes anywhere, and that will also signify that this can turn green and you can hit Analyze Data, and it'll get analyzed in the cloud. In about 15 minutes, you'll get an email with a link to your results. All right, let's go back to the presentation. So let's take a moment to talk about single contrast versus multi-contrast files. So as I mentioned, some input file types support multiple contrasts, such as CuffDiff and DE-seq. So iPathwayGuide can analyze up to 5 contrasts from a single file. Your file can contain more than 5, but you can only select up to 5 in that file. The nice thing about this is that a meta-analysis will automatically be generated. And I have a sample here of a multi-contrast file. So in this case here we have a file. This is a CuffDiff file where we have 3 contrasts, and I can choose which ones I want to have analyzed. You can see we have looks like a couple of varieties here where we have different treatment protocols and so forth. But in each of these we have about 10,000 or 11,000 genes that were identified, and then using these default thresholds we see what our DE genes are. We can look at each of these or get a preview of the volcano plot just by clicking on the view and apply. And here we have our volcano plot. We can even interrogate some of those genes and see what their fold change and p-values were from our input data. As you can see, I can't analyze the data because I haven't given it a title or a description. You can see these have been identified in red. As soon as I give it a title and or a description, then it will allow me to proceed and analyze those contrasts. So when I analyze that, and actually I've already done that, let me just go to my dashboard here. You'll see that each of those individual contrasts have already been analyzed. So here we have our untreated versus TNF, untreated versus GM-CSF, and then those 2 there. But then we also have the meta-analysis that has already been generated because this is a multi-contrast file. And I'll get into this here in just a few minutes about how to read this and what this can do for you. So now let's talk a little bit about Affymetrix cell files. So in our cell file uploader. We don't support all Affymetrix cell files. We support the mainstream ones, so the human genome U133 plus 2.0 and a few varieties therein, mouse 430, and then rat 230 as well. And there's a couple other ones. If you go to our FAQ, you'll see a list of the platforms that we currently support. We are or will be adding to these platforms in our next release that'll be coming up. When you're analyzing Affy cell files, we will do the quality control and the normalization. One thing to note, though, is that our uploader is limited to about 20 cell files, and that depends on how big they are. But that can be in any combination. Maybe you have 15 of your condition and then 5 control or something like that. The one thing, though, also to note is that you need a minimum of 3 cell files in each group, and that's so that we can assess the statistical validity. So the way that cell file uploader works is from your dashboard, when you select Analyze New Experiment, you choose your organism. Obviously, you want to choose Affy cell files. And then you just choose the files that you want to include in your 2 phenotypes here. So let me add those very quickly. And then we'll choose the control group. And then we would select upload. Now the Affy cell file uploader is quite slow, and we're aware of that. We will be working on improvements to that with the next release as well. You should budget yourself at least a few minutes for that. Again, it depends on the number of samples that you may be uploading. Here in this case, 3 versus 3, that should only take a few minutes to upload and have analyzed. I've already pre-analyzed these for this presentation. But in this case, you can see these spinners are going and they'll process for a couple minutes. Once they're done, they change to a plus. And here we can assess the quality control. In the quality control, we get to see how our housekeeping genes excuse me, scored in the QC part. And then we also have the box plots as well as density plots across the different cell files. Then, assuming that they have passed the QC, then we go into normalization, where we we're using the GCRMA normalization schema, and then you can see the density plot. In the density plot, ideally, you would like to see more or less a unified line across the different cell files. If you're satisfied with that, you would just proceed to the contrast page, and then you give it a title, a description. You can change your contrast labels as well as your thresholds for selecting DE genes. In this case, we have 20,000 genes that were measured, and we have roughly 3,000 genes that are selected as DE, just based on our default thresholds. Now, let me give you an example of a set of Affy cell files where we've rejected one of the samples. So in the QC, when we come down here, when we have this dot, may be a little hard for you to see on your screens, but it's outside of the blue band, and there's a detailed description on the left here that explains how this works. But this cell file will be excluded from through the quality control scheme. And here we've identified it here in red as which one is going to be excluded, and as well as in the density plot. So you can see which one is the one in red here that's going to be excluded. I believe it's this purple line. And but when we come down to the normalization, we now see that all of the files that are represented here are going to be included for analysis, as well as the normalized density plot. The nice thing is when we go to proceed to our contrast page, we've identified which of those samples is going to be rejected from our QC metrics here, and you can still observe these down below. One thing to point out is if you want to preserve these graphs for publication or for a paper or something like that, this is the place to do that. Once the data has been forwarded on for analysis, these charts are not available. In the updated version of iPathwayGuide that we'll be working on here, we are planning to provide these graphs after the analysis. But for right now, this is the time that you need to grab these. Okay. So let's talk a little bit about selecting for differentially expressed genes. When we're talking about whole genome or your RNA-seq workflows, generally you have 20,000 plus genes that have been identified, and you want to use a combination of both p-value threshold as well as full change thresholds. So I have an example here that I've already loaded ahead of time. And in this case, we have a little over 20,000 genes that have been measured, and using our default thresholds, we have 1,200. But let me show you how we would assess this. So when we use adjusted p-values, if we're using an adjusted p-value threshold of 0.05, by random chance alone, we should expect to see, oops, I didn't give that a value. We should see at least 5% DE genes by random chance alone. So if we have 20,000 that were measured, we should expect somewhere around at least 1,000. If our DE genes are less than 1,000, just based on that p-value threshold alone, then we really can't assess the significance of those genes because it could be just by random chance. So in this case, though, we have quite a bit more than our 1,000 or 5% as DE using this p-value threshold alone. So now this is where we would want to augment this by adding some kind of a fold change threshold. So by default, we use that 0.6, but you could choose pretty much any value you wanted. So if that's maybe a 1, maybe it's 2. The main thing is to look at it. We generally recommend that you want to shoot for somewhere between 5% and 20% as DE using both of these thresholds. In this case, I'm quite a bit less than our 5%, so I'd probably want to back this off. And I know on this particular data set that our default threshold seemed to work very, very well. So if out of the 20,000 or so genes that were measured using our default thresholds, we have about 1,200 that are DE in this case. And that would work just fine for this case. Whoops. Excuse me. Again, just I've got those metrics here. You can look at this, and here's the caveat here that if by using the p-value threshold alone, if you have less than your 5% threshold, then if that could just be by random chance alone. That's I think the main thing to take away from this slide. When we're talking about targeted panels, then the DE selection is a little bit different. P-value is less significant, and I'm sorry I forgot to put the citation here, but I came across a paper that had done an extensive study looking at using p-values for selection of significance versus fold change alone. And fold change tended to have a much stronger benefit for selecting for DE genes. So when you're talking about a targeted panel, though, that panel is already somewhat pre-biased in that sense. I've heard bioinformaticians explain to me that it's already selected for your DE genes in a sense. So their recommendation is to use fold change alone when selecting for DE genes. So in this case, we have a panel here that measured 770 genes by using a p-value threshold of, say, 1. Obviously, that gives us all 770, but then using the fold change of 0.6, that gives us 440. There's no real rule of thumb as to how many genes you should have. The one thing that you want to be aware of is that a lot of the analyses that are provided to you in iPathwayGuide are a type of enrichment analysis, and therefore you can't submit all 770 genes as your DE because you won't get anything significant on the enrichment side of things. So somewhere I would recommend using a fold change somewhere between 0.6 to maybe 1.0 or possibly a little bit higher. All right, so now let's talk about meta-analysis. I touched on this briefly. So the nice thing about meta-analysis in iPathwayGuide is that it allows you to compare your results across multiple contrasts or multiple experiments. As I mentioned, you can select up to 5 comparisons for inclusion in your meta-analysis. And you can even compare across different platforms or different data types. So maybe you've got a legacy experiment that was done using a microarray, and now you've got some new data that was done on RNA-seq. You can take both of those types of data and compare them in our meta-analysis. Or you could even include, say, proteomics data and transcriptomic data. It's, I think, a very powerful tool for you. This is auto-generated if you have a multiple contrast file. If you don't have a multiple contrast file, you will need to analyze each of those contrasts individually first and then construct a custom meta-analysis. And let me show you how one would do that here by going to my dashboard. And from the dashboard, instead of selecting new experiment, assuming that we've already analyzed all of our data, I would choose create new meta report. From here, I get a list of all the experiments that are available for me to analyze, and then I would just simply choose the contrasts that are of interest to me. And as I mentioned, I can choose from different experiments and so forth, and build up my meta-analysis. Give it a report, give it a title and a description, and then down at the bottom, just create report, and it'll automatically be generated. So I have one that's already been pre-analyzed here. So this is actually comes from a CuffDiff file with 3 contrasts. I think this is the one that I showed earlier. And you have the individual contrasts here that are available. You can interrogate them on their own. I've chosen one of the other contrasts. You can see here on the summary page some of the results here. And we have a separate webinar that goes over the entire application, so I'm not going to spend a lot of time on this part of it. But to get into the meta-analysis, we would just choose the meta-analysis from the bottom of our contrast selections. And here we have these 3 contrasts juxtaposed via a Venn diagram. And so we can choose areas that are unique to one particular contrast by selecting on that number, and the chart below, the table below, updates with those genes that are contained in there. Or if I wanted to look at the 37 genes that are in common across these 3 experiments, by clicking on the middle, I now have a table that provides that information to me. I can sort these by p-value or by fold change within any one of those contrasts. By clicking on any one of those genes, it provides me with detailed information on the right about that specific gene. So in this case here, we can see that this EREG gene is upregulated in this first contrast, but it's downregulated in the other 2. This can be a very powerful tool for identifying common traits and or unique traits across these different contrasts. And it's not limited to genes. You have the different experiments or analyses, and not to spend too much time on this, but just by clicking on pathways, I can look at the common pathways that are affected in all of these comparisons. I can even apply a correction factor. So here I have 11 pathways that have been identified, and I can even look at the rank between those. So in this case, cytokine-cytokine interaction pathway is number 1 across them, but as I go down, I can see that certain pathways have different rankings depending on the contrast that's being analyzed. The other cool thing is I can click on the title bar and kind of refocus my which contrast I'm really interested in. So this is a very powerful tool for identifying those things that are, as I mentioned, unique or common across the different contrasts. And when you're looking at a specific pathway, you see all of the DE genes and then the expression levels for them. And you can obviously interrogate that by clicking on that gene symbol and getting additional information. Okay, so that's meta-analysis in a nutshell. And as I mentioned, we do have an upcoming webinar that'll spend more time about on this particular topic. Just a brief moment here on pricing. We do have 3 pricing schemes. So we have a pay-as-you-go model where you can buy each analysis individually. The main thing to remember, though, is that iPathwayGuide is completely free to try. You can submit as much data as you want. Each data set receives its own independent trial. Each data set is viewable for 72 hours once the analysis is complete. You can share that analysis. You can get email support on it. And you only pay for it if you like the results that you see. What we find, though, is that after people have tried it 3 or 4 times, they start seeing the value in it, and they want to build meta-analyses and so forth. So we now offer a 30-day subscription, which is great if your data comes in waves, and this provides you with an unlimited access to iPathwayGuide for 30 days, gives you all of the capabilities, including the meta-analysis and our printable summary, which I didn't go over, but it's covered in our overview webinar. And it's good for small labs and as I mentioned, when you have projects where the data comes in waves. Of course, we also offer an annual subscription, which is good for 1 year, and you can analyze as much as you want. You can add additional users for really not that much additional money. You get access to any upgrades over the course of the year, and we find it's really good for larger labs and core facilities. And of course, we offer those academic discounts that everybody likes. So with that, it's a pretty brief overview of uploading data. It shouldn't be that difficult, but if you have any questions along the way, please feel free to ask us. At this point, though, if you have any specific questions now, use the question box on your GoToWebinar control panel and submit them. It looks like we already have a few in the queue. And while people are formulating their questions, I just want to let you know about a couple upcoming events. So we will be at the US HUPO conference. That's a Human Proteome Organization conference next week in Boston. We'll be guests of SciEx, so look for us in the SciEx booth. That's March 13th through the 16th. Our upcoming webinar, Generating Hypotheses Using Meta-Analysis in iPathwayGuide, is scheduled for April 5th. Look for an upcoming announcement on that. And if any of you are planning on attending the American Association of Cancer Researchers annual meeting in New Orleans, April 16th through the 20th, we will be there. So please come on by and visit us in booth 754.
Mm-hmm. All right, thank you, Andrew. So we have some questions. The very first question that we received is that, can you upload micro RNA and profiling data?
Not at this time. We do a micro RNA prediction, but that's based on either proteomics data or mRNA data. We are looking to add that capability of uploading microRNA expression data, but as of right now, iPathwayGuide does not take microRNA expression data.
Another question that we have is, do we support adjuvant microarrays?
Not at this time. We're only supporting the AFI microarrays, but if you have a specific suggestion of some adjuvant arrays that you would like to have supported, please send us an email and let us know about which ones you would like, because we will be adding some additional microarray support. So if the user community wants a specific one, we'll take a hard look at adding that.
Another question that we have is, if the user has data, RNA-seq data from different cell lines, say 10 different cell lines, each, for instance, has a treated and untreated data. I'm guessing these are drugs. What would be the best way to examine the effect of the drug across cell lines?
I think the main thing would be to analyze each one individually, first of all, and make sure that you like the results on each one of those, but then use our meta-analysis. I think that's a really powerful tool for comparing effectiveness of therapeutic response on those, if you're targeting the right pathways and or the right genes, and it's a great way to compare and contrast each one.
There is actually I wanted to add to this, there's actually a webinar that's posted on the Advaita Bio website, which deals with contrasting therapeutic response. I use public data to analyze it, but you probably just want to take 5 of your cell lines and treated versus untreated, and then analyze the response for that. The meta-analysis is actually great for comparing cell lines or any kind of therapeutic response between cell lines.
Right. In fact, I've got our website up here. Just you would click support and then choose iPathwayGuide, and then you'll see down here in the webinar section on the left-hand side, if you click that, we've got a few different previously recorded webinars that are available. And I think this is the one that you're mentioning here, Sidra, leveraging public data, comparing therapeutic response in melanoma. So this is a really good webinar that would show you how you would do that.
Thank you, Andrew. And then another question we have is there any method to segregate genes based on cell fate function, like differentiation or proliferation, it's just cell fate function, from the list of DE genes?
I'm not aware of that capability in iPathwayGuide.
No. So. You have to upload the list, the total list of genes, and then the analysis happens in which, based on cell fate function, you're probably looking for gene ontology terms per se, but you are not going to be able to, you're basically doing manual segregation if you're going to do a segregation yourself, and you're biasing the results of your analysis based on that.
Right.
Okay, one more question that we have is in DE-seq2. Fold change is related to order of the contrast, alphabetical. Is there a way to change the fold change value that is plus to minus inside, like reversing the contrast, as in fold change positive to negative?
Gotcha. Well, first of all, that sounds like it's more of a question specific to DE-seq. I will say that we've run into some issues with DE-seq because it's not a strongly structured format. So I think you'd have to go back to the DE-seq documentation and look at that. We don't support the use of DE-seq, but the output from it, if you follow their general guidelines, we can support that. I would recommend, if you need to reorder some things, go back to their documentation and see if you can do it that way.
Thanks, Andrew. And another, just one of the last few questions is there a way to do single cell clustering analysis using single cell RNA-seq data?
Not today. We are working with a group on specifically about single cell analysis and looking at how you would look at that, but it's a different type of workflow with some specific elements that need to be considered. So we are working on that, but it's not in our application today, unfortunately. But it's coming. We know that single cell is definitely a growing area of interest, and people would like to be able to do some of that. So.
Another question is it possible to reconstruct pathways based on the analyzed data?
That also will be coming up in our upcoming release. We'll have network construction, as well as we've got a whole list of things that we're going to be including in our next release for iPathwayGuide, and one of them is the network construction based on the whatever genes that you happen to select, so you can see how they're related.
Okay.
I think we have time for one more.
Yeah, we have time for one more. If I want to change thresholds after my data has been analyzed, do I need to resubmit my data?
Yeah, actually you do, because it becomes a different set of DE genes based on that, and because of our pathway analysis where we assess the perturbation of the pathways, it's not a quick and dirty type of calculation like enrichment is. So for our advanced method for analyzing the pathways, you do need to resubmit those data. In our next release, I think we'll have it so that your input data is preserved, so you can just call that input file back up and then just change the thresholds, and then you're off and running. So it'll save you some upload time in the next release.
Just before we, I think, are pretty much all set with the questions, but could you possibly just go over the subscription options again? Because one of the attendees wanted to know that if each cell line is considered a separate project, so if it's going to be a pay-as-you-go option, or if they can just, upload as much data as you'd like in the 30-day and the annual subscription. If you can just go over this really briefly.
So again, in the pay-as-you-go model, each data set is an independent analysis. So now it depends, though, if you're using CuffDiff or one of these other file formats that supports multiple contrasts. That file, or I should say the 5 contrasts, up to 5 contrasts that you select from that file are considered a single analysis. But each if you have to submit, say if you have 10 cell lines or something like that, each has its own data set, that would be 10 data sets. Each one of those has a preview, and you can do that as many times as you want. But I think if you've got more than a handful of data sets, it just makes sense to just get a 30-day subscription and just analyze to your heart's content, or go to an annual. I mean, but at a minimum, you'd want to do a 30-day.
All right. I hope that answers your question. And I think that's about it for questions for now.
Okay.
And there are some more questions, but I will email people back with the answers for the specific questions because they seem to be very user specific. And we look forward to seeing you guys in the next webinar on April 5th or at any one of these trade shows that Andrew mentioned. All right.
I'll make the recording available. It should be up probably later today, tomorrow at the latest. You will get an automated email for it, and then we will also post it to our website as well as YouTube. So you'll have plenty of access to hit this again. And if you have any individual questions, follow-up questions, feel free to contact us. Just use the contact us button from our website or from within the application. There's a feedback button at the top right corner. Click on that, and Sidra or I will get back to you just as quickly as we can. So with that, thank you, everybody, and have a great rest of the day. Bye.